Hey learners, how are you all? Welcome and welcome back to the part 6 of this lecture, chapter 8, Cell the Unit of Life. We are done by the major part of the cytoskeleton elements, and there are two types, the microfilament and the microtubules. In this part, we'll focus on the intermediate filaments, while we'll also talk about the centriole and centrosomes with their functions, and we'll end this lecture by the nucleus. So now let's begin by the centrioles first, and these are the above mentioned subtopics which we'll focus on. Now let's begin. So the centriole is present in all the eukaryotic cells, found mostly in the animal cells and some protista, but they are not in the higher plants and fungi. It is cylindrical shaped structure, or more specifically you can say barrel shaped structures. Do you know wine barrel cans? Yeah, so it is similar to that, with the polarized ends made up of the microtubules. Unlike the cilia and flagella, it has 9 triplet microtubules with the 0 singlet in it, as it has 9 plus 0 arrangement in it. Like the basal body and the centriole, they both have the 9 plus 0 arrangement in them. So as I said in the previous lecture, that the basal body and the centriole are two main microtubules organizing center, where the centriole always lies near the nucleus, so it is also called as the nucleus site of the cilia and flagella, and that is why the centriole is responsible to form the basal body. Don't be confused. The basal body and the centrioles are the same structure, but with the different in their functions and positions. Right? See, they are interconvertible. It means that the centriole can function as the basal body, but the basal body cannot. And their another function is in the animal cells. The centriole plays an important role in the cell division by arranging their spindle fibers between two poles of the cell. The location of the centriole during the cell division decides the panel of the division, and the panel division is always at the right angle to the spindle. Let's see what I'm saying is. Now let's see their structure first. So in the animal cells, a pair of centriole is present, which is known as the centrosome, where two centrioles, one is the centriole is termed as the mother centriole and the another termed as the daughter centriole, are arranged perpendicularly to each other in the center of the centrosome. And the cloud amorphous material that surrounds a pair of centrioles is called the centrosome matrix, or you can also say the pericentriolar material, PCM. Let me use a more clear diagram for you. So these are the short microtubules like structure radiating outwards from the centrosome in all the directions to form the aster. So the aster is basically a star-shaped array of the microtubules. The word aster comes from the Greek word meaning star because of its radial appearance. You can see in the diagram. If you look it under the microscope, you will see a sunburst or star-like structure similar to that. And these asters forms in the early stage of the cell division, and it functions in the spindle positioning and to ensure the proper chromosomal segregation. You will see in the higher classes about this. So you can see the formation of asters only during the meiosis and mitosis. So what happens is, when the pre-existing mother centriole acts as a scaffold to a new daughter centriole, or you can say procentriole, to form a 90-degree angle to the axis of the mother with the amorphous thick liquid or the aster-like structure. Together this leads to form the complete centrosome organelle, which is located just outside the nucleus. The mother centriole has the appendix and can form the basal body out of it, and the daughter centriole is newer, so they lacks this appendix. So during the cell cycle, the centrioles duplicate once per year to ensure each centriole can inherit a pair. So the distal and subdistal appendages mainly found on the mother centriole, the older one of the two centrosomes, where the distal refers to a position away from a point of origin, and while the subdistal, or you can say proximal end, refers to the position which is slightly like just below the distal end. Like you can see in the diagram how the distal end is located at the very end of the mother centriole, marks the start of the exome. That is why we also call it the basal plates, and involved in attaching the cell membrane during the celiogenesis. That's the another process. and the proximal end is located on the other side of the centriole and are involved in the microtubule anchoring and the formation of the mitotic spindle fibers. And even you can see this cartwheel like structure at the proximal end of this immature daughter centrioles, or you can say the procentrioles. Later on, as the centriole matures, their cartwheel structure gradually disappears or diminishes. It becomes the hollow cylinder made up of the microtubules triplet only. So now let's see how their mature structure looks like. Centrioles are the self-replicating units. You will see more about their nature and their procedures in your higher classes. For now, let's just move by the next slide. Let's see the transverse section of the centriole. Centriole is a highly structured and symmetrical organelle which exhibit a cartwheel structure in their transverse section. The centriole has 9+0 arrangement in them. Now let's break down their structure in a little more detailed way. So the centriole consists of 9 evenly spaced peripheral triplet fibrils of microtubule arranged in a ring. You can count it. This is the 1, 2, 3, 4, 5, 6, 7, 8, and this one is 9. So each triplet consists of 3 tubules in them, which are named as A tubules, B tubules and C tubules, where the A tubules has the complete microfilaments in them. It means 13 protofilaments, where the B tubules has incomplete 10 protofilaments in them, and the C tubules has the again incomplete 10 protofilaments in them. They partially overlaps each other's structure, like the B attached to the A and in the same way C attached to the B. Like you can think it of your own. Making all 3 complete tubules would waste a lot of energy and space. We know that the centriole walls needs a rigid framework, not a complete tubule structures, right? Just to save the resources, these tubules shows their lateral sharing with the others, where the C tubule doesn't participate directly in the exome formation. The only A and B continues to the flagellar shaft, so it doesn't need to be complete. It mainly anchors and supports the triplet structure in the centriole wall. So these 9 triplets form a symmetrical ring with no as such central singlet in between. Unlike the cilia and flagella, it has 0 central pair in the center. Other than the central microtubule, it has a central hub in between, which is a rod-like proteinaceous structure, which is connected with the peripheral triplets by the radial spokes. You know what radial spokes is? Radial spoke is again a proteinaceous structure. So these radial spokes extends outwards and connects to each of the 9 triplets, like the spokes on a wheel. It is consist of total 9 spokes for each triplet in it, and then these triplets are interconnected or linked with the help of the AC linkers, also called as triplet linkers or the intertriplet bridge, where the A tubule of one triplet is connected with the C tubule of the next adjacent triplet, that is by the protein connections that links them together is called as the AC linkers or the AC bridge, which connects these neighboring triplets together and to give a more stabilized structure. Like if you count it the clockwise direction, then you can say AC linkers, and if you count it in the anti-clockwise counting, then you can say CA linkers. That depends on how you count it. So together the central hub, radial spokes and the triplets, they form a cartwheel like structure, which you can clearly see in the immature newly formed centrioles. Then we have the pericentriolar satellites, which is present on the outside all around the centriole. They are the small electron dense granules floating on the cytoplasm near the centrosome. That is why they are called as the pericentriolar satellites, or also you can say mesiule or the centrosphere. These are the older terms, but in nowadays we use PCM to be specific. Keep this thing in your mind that they are not the part of the centriole itself, but they just lies around the periphery of the centriole. You know how the satellites floats around the space? It's like similar to that, it floats in the space. Like the satellites orbiting a planet, these protein granules orbits around the centrosome, like similar to that, as they are made up of proteins. So while moving along the microtubules, their main function is to deliver these regulatory proteins to the centrosome, which are needed for the microtubules, and they also help in the assembly of the basal body and the cilia. Unlike the continuous pericentriolar material PCM, which we know already, that directly surrounds the centrioles, these satellites are different. These satellites are separate granules that can also be found distributed throughout the cytoplasm.
Centriole is a membraneless structure. It has no plasma membrane on its structure like the cilia and flagella do have.
And the microtubules are the highly dynamic structures. They are constantly assembling and disassembling by the process called the dynamic instability. And other than that, if you see, the rest of the structures are similar to the structure of the cilia and flagella. So yeah, that is it. Now we have some of the other important functions of the microtubule, as we done by the one of their important function, which is the locomotion in the cilia and flagella. In detail, that how the microtubules contribute in the formation of some structures like the cilia, flagella, and the centriole. And we are done by their functions according to their structures. Now we have some of their other functions like the structural support. So let's see them. They are rigid hollow tube like structure. They act like the scaffold or the support beams inside the cell, and they help the cell to maintain their shape and to resist the pressure. Their hollow tube like structure actually helps them to resist the external pressure and to act like the scaffold. Without the help of it, the cell can collapse into a thin sheet. Their rigid framework resists the compression. For example, in the nerve cells, their long axons are supported by these microtubules to prevent them from bending and also from the breaking. They are also involved in the intracellular transport. They act like the trackers, or you can say highways, for the movement of the cell materials. So you can also call them cargo highways. There are two of the special proteins named as kinases or dynein. So you will see them in your next year. And these microtubules provide the track for the motor proteins so that they can move the organelles like mitochondria or vesicles along with them. For example, in the nerve cells, the vesicles move along with the microtubules to different parts of the cell for their signal transmission. There another important function is cell division or spindle formation in the basal body or the centrioles. During meiosis and mitosis, the microtubule forms the spindle fiber that helps to separate the chromosomes equally into the two daughter cells. For example, in the human cells, the microtubules ensures their equal distribution of their chromosomes to the daughter cells during the mitosis. There are also some of the drugs that can affect the microtubules and the actin filaments. Like these cytoskeletal elements are the target of a variety of drugs, if you know. So that act by the interacting with their polymerization and depolymerization of their subunits. For example, some of the common drugs that can affect these microtubules and the actin filaments is colchicine. Actually, colchicine is a mitotic poison which can inhibit the microtubules and can easily arrest the cell division in the cell. So what if any human consumes the colchicine chemicals? What will happen to his body? So first, the colchicine binds to the tubulins, the protein that makes up the microtubules, and this prevents the microtubule polymerization, meaning new microtubules cannot form further. So without the microtubules, the mitotic spindle cannot develop. As a result, the cells cannot divide and can get in the metaphase. It can affect the rapidly dividing cells the most because they need the mitosis continuously to be happen. So due to the intentional cell death, it can lead to the nausea, vomiting, and multi-organelle failures as well. There are some of the other famous examples of the drugs like vinblastine, taxol. You will see these examples in your higher classes, correct? And at the end, microtubules are also involved in the positioning and the organization of the organelles. within the cell. They can position and can organize the organelle by providing them the structural support. Basically, microtubules decide the position of the organelle and cell polarity so that the cell can decide their position and can orient themselves within the tissue by their own self. For example, Golgi body, ER, mitochondria within the cytoplasm, they can arrange in the cytoplasm. That which side of the cell faces where. For example, we know that the Golgi apparatus is fixed. Their position is fixed near the nucleus. By what? By with the help of these microtubules only. And in the plant cells, these microtubules guide the cellulose deposition and their shaping of the cell wall. You will learn about it in the cellulose biosynthesis in the plant cells. And the microtubules also function in the signal transmission, where the microtubules helps in the signal transport by influencing the cell signaling pathway. Let's keep it for some other chapter. We'll learn in the cell signaling part. Then we have the third and the last type of our cytoskeleton element, which is called as the intermediate filaments or IFS. These filaments has the diameter somewhere in between the microfilaments and the microtubules. It is about 8 to 12 nanometer in their diameter, or you can also consider 10 nanometer in a standard way. Unlike the microfilaments and microtubules, these intermediate filaments do not participate in any kind of cell mortality, so that is why it is not given in your NCERT. So it is up to you that you either want to do it or not. If not, then you can just skip to the next part, to the nucleus. So now let's begin by this. So these filaments are found in all the eukaryotes but not in the plants and fungi. Unlike the actin and tubulin proteins, these intermediate filament proteins are chemically heterogeneous and can show tissue as well as the specific species variation in their molecular weight. See, we know that the actin filament is made up of the actin, which is a single protein compound, and the microtubules are composed of the tubulin dimers, alpha and beta tubulins. But the intermediate filaments are made up of the various proteins depending on their cell types. There are even more than 50 different types of intermediate filament proteins have been identified yet and classified into the 6 group based on their sequence and the tissue distribution, which we will see by now, where each type of IFS protein has a similar overall structure in each of them but vary differently amino acid sequence and terminal domains. Hence, they make them chemically heterogeneous from the others too. But before going to their protein classification, let's see their structure first. So the intermediate filaments are the polymer composed of alpha helical monomers assembled together just as a rope is made up of many smaller strings. Similarly, the intermediate filaments are made up of many thin polymers joined together. First, we have the single polypeptide of the intermediate filament. It's an elongated molecule consisting of 3 domens on it: head, rod and the tail domain respectively. At the end terminal, it has non-alpha helical head domain, and at the central part it has alpha helical rod domain, and at the end, at the C terminal, it has again non-alpha helical tail domain, where the central rod domain consists of a long tandem structure which repeats one behind the another of a distinctive 7 amino acid sequence, which is called as the heptate repeats. So let's see what are the steps we need to follow. First, in the step 1, when the 2 polypeptide chain of monomeric IFS protein parallelly coins together to form a dimer with an asymmetrical head and tail. Then in the step 2, these 2 dimers stacks together in an anti-parallel way to form the tetramer, which is a 4 polypeptide chain, looks like a rod-like structure. And then in the step 3, these 2 anti-parallel tetramers joins together to form the protofilament. Then the 2 protofilament associates to form 1 protofibril. And then in the step 4, similarly the 4 protofibrils joins together to form a short thick unit length filament called the ULF. So the ULF has reached the approximate final filament diameter, but it is still shorter than the mature filament. So we can consider the ULF as the first morphologically visible IFS intermediate filaments assembly seen till now in the complete arrangement. Then in the last step 6, the several ULFs arrange end to end longitudinally, and then the compact radially final 10 nanometer intermediate filament is formed. These filaments rapidly grow in their length, and finally the mature rope-like intermediate filament is formed at the end. Then there is a proper coiled coil model of intermediate filament is given alongside, so you can move side by side. And one thing I forget to tell you at the second step, that the dimers are anti-parallel, their tetramer has no structural polarity on them. Unlike the actin and microtubules subunits, the intermediate filament subunit do not contain a binding site for them for any of like the ATP or ADP units. So the anti-parallel arrangement of their dimer implies that the tetramer, and hence the intermediate filament that is formed, is a non-polarized structure. So if we talk about their types of protein, we have more than 50 different types of IFS proteins discovered till now, but we are going to classify them into only 6 groups based on their sequence and their tissue distribution. So let's see what are these 6. So first, in the type of IFS protein, first type is the acidic keratins, and the type 2 is the neutral or the basic keratin. These are the two types of complementary keratin proteins, and they are present in the epithelial cells in a particular ratio. They always comes in a two complementary types. I mean, the type A acidic keratin always pair with a type 2 basic keratin. Together they form a coiled coil heterodimer, the basic subunit of keratin IFS filament. This pair is obligatory. It means that neither the acidic or basic keratin can form the stable filament alone. One acidic and one basic keratin, this combination becomes essential for the correct filament assembly and their mechanical strength in the epithelial tissue. In any of the epithelial cell, their opposite charges balance each other and stabilize the coiled coil structure together. Now the keratins can be either hard keratin or the soft keratin. Their particularly main function is in the formation of any kind of hard structures like the hair or nail. These are like the hard structures formed by these hard keratins, where the hard keratin mainly function is to provide the strength and rigidity to these structures, while the soft keratin, they are generally present in the cytoplasmic region of the epithelial cell, and their main function is to provide the flexibility and the protection to the areas like the outer layer of the skin. For example, in the hair or nails, the acidic keratin contribute to the hard keratin in the hair or nail, and the epidermis forming the tough protective structure which cross-linked with the basic keratin. Then comes the type 3 vimentin. Vimentin is the most widely distributed of all the types of IFS protein, which is typically expressed in many of the specialized cells like the leukocyte cells and the blood cells and the endothelial cells and some epithelial cells and mesenchymal cells such as the fibroblast and many more. It has few different subtypes like the desmens is present in the muscle cells like the skeletal, cardiac and the smooth muscle cells. Their main function is to maintain the cell shape, anchor the organelles and to intact these organelles at their position like the nucleus and the mitochondria. Then comes the neurofilament type 4, which is generally expressed in the mature neurons. They are generally present in the axon of the neuron to provide them the structural support and to maintain the axon diameter and to help them to determine the nerve conduction speed. Then comes the most important type 5 nuclear lamins. These lamins are found exclusively in the nucleus. These lamins form a network-like structure that lines the inside surface of the inner nuclear membrane and termed as the nuclear lamins. They are present in the nucleus of all the eukaryotic cells, and this is one of the most frequently ever asked question in all of the exams. They function as to support and to maintain the nuclear shape and their chromatin organization. Then comes the last, our sixth type of protein called the nestin. Nestins are generally found in the stem cells of your central nervous system and in the developing tissues. Moreover, they provide the structural support in developing neurons and the muscle precursors and to replace by the other IFS like the neurofilaments or the desmins as the cell matures. For example, they are in the embryonic brain and the spinal cord and also in the developing skeletal muscles, as they have the similar role like the alpha internexins do have. Now let's see what are the various functions performed by the IFS filaments. Like we have few of them. Like they provide the mechanical strength. As we all know that the IFS are very strong and flexible like the steel cable inside a cell. So they prevents the cell from any kind of breaking or stretching under the stress. As we saw, keratin is in the skin cells which helps the skin to keep away from any kind of tearing, and the desmins in the muscle cells prevents the muscle fiber from any kind of damage during the contractions. Now suppose as we all do exercise in the morning, pull-ups or stretching your legs, your skin is facing a lot of stress forces, right? Now what if these IFS filaments are not there in that cells? So what will happen then? If these IFS are not paired, the stretch would create breakage in the epithelial cells that can even harm your skin. Cells with the intermediate filament can resist the tension and can remain intact with each other, while the cells which don't have the intermediate filaments in them, they can rupture easily. IFS also gives cell junctional support that you can consider as an another important feature of it. IFS connect cell to cell and cell to matrix attachments where we call desmosomes in cell to cell junctions and hemidesmosomes in cell to the basement junctions. They give the tissue, the skin or any epithelial cells a very tensile strength. For example, the keratin links the skin cells tightly so the skin doesn't peel apart. Now you know why the presence of the normal keratin is essential for the mechanical strength to the epithelial cells. Now there is a very rare genetic disorder called the epidermolysis bullosa based on the similar problem. Experimental evidences in humans has shown that the absence or expression of the mutual keratin, in which the mutant keratin inhibits the assembly of the keratin filaments where the keratin cannot form the proper diamonds, which leads to the defective or the incomplete filament assembly, leaving the cells structurally weaker, results in the several skin abnormalities. EBS is one of them. This can be seen usually in the children at the time of their birth itself. So the expression of the mutant keratin is in the cousins when the cousins get married, which is called the consanguineous marriage, when the parents are carrying the same genes to their children. You know, there is always more chances of recessive genetic disorder in the kids who have the similar genes to their parents, and in the case of cousin marriages the chances become really high as they are inherited from a common ancestors. That is why in the India, like in the northern part of India especially, the marriages in the same gotra is not allowed. Epidermolysis bullosa is not limited to children's only. See, it's a genetic disorder, so people usually born with it, but symptoms can appear at any age depending on the severity of the mutations. If we talk about their symptoms, it is observed that any of the slightest of the injury, being by rubbing, scratching, or even by the touch of any hard surface, causes the lysis of the epidermal cells, which can lead to the skin blisters and can breaking in the skin and the formation of fluid filled bulla, and even in the severe cases this can cause the death of the child as it has no cure. You can see these bubbles on the hands. Sometimes these bubbles are clear or maybe they are filled with blood. Sometimes the people might not realize themselves as the condition can change daily. Some days the skin looks fine and the next day new blisters appear suddenly. So you know it becomes very hard to treat them. They are not just on hands, but the blisters can occur anywhere on the body where friction can occur, like on the elbows, knees, shoulder, under thigh or sometimes even inside the mouth or some, and the another function of IFS is to maintain the cell shape and their integrity. A very common word used to describe this is the cytoarchitecture, which means the structural organization of a cell, that how the internal framework, which is mainly the cytoskeleton elements, maintain its shape, stability, and their internal arrangement in it. They form a network beneath the plasma membrane and around the nucleus, which is called as the nuclear lamina. This supports the overall cell shape and to maintain their internal structure. So the word cytoarchitecture in the neurofilaments suggests the structure must be strong and flexible at the same time, and that is where the neurofilament comes in. Neurofilaments are basically a special type of IFS filament found only in the neurons. They form the internal support system of the nerve cells as they are part of the cytoskeleton. Along with that, it has microtubules for the transport and actin for the shape and movement, you can see in the diagram. And they anchors means helps to position the organelle, especially the nucleus, like holding the nucleus in their place. For example, vimentin filaments keep the nucleus and the mitochondria properly arranged in the connective tissue. They provide shape, strength, and support to the nuclear envelope in organizing their DNA. In the upcoming slides, we will learn about the nuclear envelope in detail. See, one thing I would like to clear here, that the IFS are involved in the cell migration and movement. No doubt that the actin and the microtubules are the main engine of the cell mortality, but the IFS play only the supporting or the coordinating role that makes movement of the cell. See, when a cell moves, it changes its shape constantly by extending at the front, which is the leading edge, and the retracting at the back. IFS provides mechanical stability during the movement by prevents the cell from tearing apart as it stretches and squeezes through the tissue. For example, in the vein blastine, the IFS fibroblast protect the cell from getting ruptured during the cell migration. IFS interacting with the actin and microtubules which guide the direction to the IFS, and together they make sure that the cell moves in a coordinated and in a polarized way with a clear front and the rear end. I am repeating again, the intermediate filaments don't push or pull the cell forward, but they just make sure that the moving cell stays strong and coordinated and protected during their movement, their migration. Correct? Then the another function of IFS is signal modulation. Yes, they actively regulate cell signaling pathways that controls many things like cell growth, their shape, their stress response, and migration. IFS sends mechanical stress like the stretching, compression, and they convert it into the biochemical signals. Correct. They transmit these signals to the nucleus or to the other organelles. For example, in the epithelial cell, wait, give me a minute. Sorry for that sound. So I was saying that the IFS controls the complete cell behavior by the signal modulation and decides where and when the signaling can occur. So basically you will learn more about it, these signals, in your graduation. So that is enough for now. The last function is in the cell type identification, as they are used in the medical diagnosis. See, each cell type has a unique IFS. They are used as the biomarkers, or you can say cell ID markers in the pathology in the labs. For example, these are the examples, you can check them out. Now we have the another topic, nucleus. Perfect. Now let's see. So basically the nucleus is called as the brain of the cell or the controller of the cell. Just like our brain controls all of the body activities, similarly the nucleus controls all the cellular activities inside it. Just like our brain stores and pauses the information in it, similarly the nucleus stores the DNA, which is the genetic material of the cell. So the nucleus stores the DNA and processes the genetic information, similar to our brain. Nucleus is singular and nuclei is the plural of the nucleus. If you talk about their discovery and shape, so nucleus is a round and dense cell organelle
which is discovered by the Robert Brown as early as 1831. It is a double membrane bound organelle. Generally, if we talk about the number, every eukaryotic cells possess one or more nuclei during their life cycle at some stage of their existence. Generally, the eukaryotic cells contain at least one nucleus in them, and they are named as uninucleated. And most of the eukaryotic cells have many nuclei in them, and they are termed as the multinucleated. And along with them, some of the eukaryotic cells also have zero nucleus in them, and they are termed as eunucleated. For example of eunucleated cells, there is mature sieve tube cells and mature erythrocyte cells. See, what happens is in the mammalian RBCs, they have nuclei when they are young, but however when they lost it when they are matures. And similarly, in the flowering plants, the sieve tube cells of the phloem, they are involved in the conduction of their foods. Also, they also lost their nuclei at the time of maturity. So the number of nuclei also varies, correct? There is also some multinucleated conditions has seen in the flowering plants, but remember, more than one nucleus may be present in the lower organisms usually. For example, in the tapetum, which is in the anther of the flowering plants, and as one nucleus is called uninucleated, and similarly when the cells has two nucleus, it is called as binucleated, then trinucleated, and so on. So now tell me, do you know what is dicaryons, and is there any difference between the dicaryons and the binucleated cells? Tell me if you know that. If any one of you is thinking that there is no difference between them, they both are similar, the dikaryon and binucleated, then let me clear you, no, they both are not similar. They refers to a similar thing but in a different context. See, the binucleated means two nuclei from the same parent, but the dicaryons means the two nuclei from the two different parent nucleus. Dicaryons is in the fungi and it has two distinct nuclei. Similar of the multinucleated, it has multicaryons or multikaryotic cells. It means that many nuclei from many different parent nucleus. That is again in the fungi. You will learn more about it in the biological classification chapter. So for now, let's see what we have next. Now cell division involves two stages, or we can say cell division phases. One is the interphase and another is the M phase. In the interphase is the period of cell growth and preparation of their division. And in the M phase, which is M stands for the mitotic phase, where it's a period where the cell actually divides. Now we will see how the nucleus behaves in the cell division cycle in both the interphase and the M phase. So basically in the interphase, nucleus is clearly visible, and that's what we are going to study in this. So the interphase is the longest phase of the cell cycle, where the cell grows, duplicate its DNA and prepare for mitosis for the M phase. So the interphase is basically, it has sub phases. Just as in the humans, they have different life stages like the childhood, then teenage, then adulthood, then they gets mature and creates the next generation. Similarly, a cell's nucleus has sub phases. They grows, they prepare themselves for the division and give rises to the new nuclei by continuing the cycle of life. During the cell cycle to the stages of human life, each phase has its own personality, activity and purpose, just like the stages in the lives. If we see the stages in the interphase, it has the G1, S and the G2. You can call these three as the three stages of the interphase, where you can clearly see the nucleus under a microscope and the chromatin material, the uncoiled thread-like extended form of DNA, which is not yet condensed into the chromosome. That chromatin you can see in this phase. The nucleus is functional and clearly visible because the nuclear envelope and the nucleolus are present in it, and the chromatin is loosely packed. And later on, when the cell enters in the M phase, the nucleus is not clearly visible as of the mitosis, the nuclear division. This is when the nucleus disappears temporarily as the cell divides. So Again, it has four stages: the prophase, the metaphase, the anaphase, and the telophase. These are the four stages of the M phase. The nucleus begins to disappear in the prophase, to be specific, and the nuclear envelope starts to break down while their chromatin condenses into the visible chromosome and their nucleolus finally disappears. You will see in more detail that how the nucleus visibility changes with the M phase of the cell cycle in the cell cycle and cell division chapter. For now, let's see the nucleus in the interphase. So overall, you can say in a cell where the chromosomes are visible, the nucleus is not visible, and vice versa, in a cell where the nucleus is visible, the chromosomes are not visible. So you can either see the nucleus or the chromosome at a time, but not both at the same time. Correct? Now we have the structure of the interphase nucleus. We'll cover this topic in two different parts. One is the nuclear envelope and another is the nucleoplasm. So let's begin by the first part, which is the nuclear envelope. The nuclear envelope is the covering of the nucleus which provides the mechanical stability to the double membrane system. So the electron microscopic study has revealed that the nuclear envelope, which is consists of the two parallel membranes, one is the outer membrane and another is the inner membrane. This envelope forms a barrier between the materials present inside the nucleus and that of the cytoplasm. So let's see what is the outer nuclear membrane. So on its double membrane organelle on the outer side, you will find the outer membrane, which is usually remains continuous with the endoplasmic reticulum, and more specifically the rough endoplasmic reticulum RER, and also this bears the ribosomes on it. Ribosomes attach to its surface just like the rough ER do have. Now it helps in the transport of the materials between the nucleus and the cytoplasm as it lies on the outer side of the membrane, and it also provides a connection between the nucleus and the cytoplasmic membrane system. Now it has the another membrane called the inner nuclear membrane. It lies just beneath the outer membrane and faces the nucleoplasm. Nucleoplasm is the fluid inside the nucleus, and it is supported by a network of fibrous protein structure called what? You know that? It's called nuclear lamina. Now this nuclear lamina provides the mechanical support and also maintains the shape of the nucleus, and also it anchors the chromatin, which is the DNA protein complexes inside it, and the nuclear pore complex, which I will explain in the next slide. Now I hope both of these two concentric unit membranes are clear now. They both are different from each other on the basis of their structure and function. The outer nuclear membrane is different from the inner nuclear membrane on the basis of their chemical composition and the functions they perform. Let's see them. We saw how the outer membrane is in the direct contact with the ER, but the inner membrane is not, and simultaneously they have, as per their structure, they have different functions to perform, and their membranes of both the organelles like the nucleus and the ER membrane are similar in their structure and chemical composition. The thickness of both the membrane is similar, and they both provide the site for the attachment of the ribosomes. However, no ribosomes are attached with the inner nuclear membrane because it is closely associated with the chromatin materials. But before that, there is another space between these two layers called the perinuclear space or PNS. This also called as the perinuclear cisternae, as it is the space or the gap between the inner and the outer membrane of the nuclear envelope. See, as I said, the outer nuclear membrane is continuous with the ER. Therefore, the perinuclear space is continuous with the lumen inside of the ER. Okay, so this means that the materials like the ions and the small molecules can move freely between the ER lumen and the perinuclear space. So yeah, as you can see, the diagram we have already done by the outer nuclear membrane and the inner nuclear membrane combinedly called the nuclear envelope. Then we saw the nuclear lamina, the structure formed by the filaments of the lamine protein, and we saw the perinuclear space in between them, and we saw the RER and the ribosomal structures on them. One more thing, the width of this PNS space varies between the 10 nanometer to 50 nanometer. This is the space, this is not the gap. The gap is the different thing, that is the pore, that I will explain by now.
Now let me clear you one thing before starting, that the topic nuclear pore complexes or the NPC is not given in your NCERT. So only if you are a graduate or the undergraduate student can watch further. If you are not, then please I request you to skip this part. So now let's begin. See, the nuclear envelope is not continuous. A number of pores are present in it through which the nucleoplasm is connected to the cytoplasm. These pores are called what? Nuclear pores. And at the nuclear pore sites, only at the nuclear pore sites, the outer nuclear envelope is continuous with the inner nuclear envelope. So, at a number of places, the nuclear envelope is interrupted by minute pores which are formed by the fusion of two membranes. This is your outer membrane and this one is your inner membrane, and in between it has the perinuclear space, and this rest of this complete cylindrical structure is nothing but your exact NPC. It is like an opening between these two membranes which helps to transport of the molecules in and out and again out and in by connecting through this nucleoplasm to the cytoplasm and again vice versa, cytoplasm to the nucleoplasm. It controls the exchange of the material between these two, but not all the materials, only the specific molecules between the nucleus and the cytoplasm. You can see the directions. The import means into the nucleus and export means out of the nucleus. For example, in the import of histone proteins, DNA and RNA polymerase, splicing proteins and transcription factors and many such different things. Histone proteins are formed in the cytosol and they get imported into the nucleus for the packaging of the DNA, if you know that. And the transcription factors regulates the gene expression. We'll learn more about it in the molecular biology. Other than the proteins, the nucleotides and enzymes also moves in, and in the export of the materials like RNA and ribosomal subunit moves out of it. These pores actually regulates the traffic between the nucleus and the cytoplasm, but not through the active ion pumping, like any of you thinking about the active ion pumping or the protein pump structures, then no. The nuclear envelope doesn't have these typical protein pump-like structures, those who found in the plasma membrane and like sodium-potassium pumps. However, it doesn't have these specialized transport proteins embedded in its structures. They don't need it as they are itself made up of dozens of protein, which I'll explain in further slides. So the nucleus doesn't have protein pumps, but it do have the protein based transport systems like the nucleoporin, importin, exportin, which allows the controlled movement of molecules using energy, GTP, used in a different way. And for these protein pumps it is ATP based, we already know that, and in these NPC their nucleotide binding site is GTP. So make sure you'll not confuse in these topics. And these protein pumps, now you know why there is no traditional protein pumps in it. Like the ions and pump or the protein pumps are mainly used in the membranes that control the ionic gradient or the membrane potential, which is required in the plasma membrane, in the mitochondrial membrane, in the chloroplast membrane, or in the lysosomal or the vacuolar membranes, but not in the nucleus, right? So these nucleus envelope doesn't maintain any large ionic gradient across it. Instead, its main job is to do the selective molecular exchange, not the active pumping of the ions. If any of you is still doubtful, I'll clear in the next slide. But before that, let's see their basics first. If you see their overall size, it is 120 nanometer in its diameter, which makes it one of the most largest protein complex structure in the cell. It is 30 times more larger than the structure of a ribosome, a typical ribosome. Nature defined everything on its way so that the ribosomes, then their subunits, can easily pass through these pores. If you see their number, it is present in the multiple copies like 300 to 400 nuclear pores per cell, and this NPC is guarded by an octagonal discoid structure of the nucleoplasmin protein. It has a symmetrical structure with an octagonal, means 8-fold radial symmetry. You can see in the diagram the electron microscopic structure of the nuclear membrane showing the nuclear pores. You can see this arrow. Like suppose if we take this single NPC and we study what's inside it. Okay, let's see this how. Let me take a blank page here. You can see a dense region. This is the cross-sectional view of the pore complex. In the B part, if we zoom in the structure, we can see the top view facing the multiple NPCs seen from the cytoplasmic surface by using the electron microscope, where each pore shows a distinct 8-fold rotational symmetry, where these 8 identical subunits or the radial spokes are arranged in a ring. You can count these labeled numbers 1 to 8. Can you see this central hole in the center of this image? Look at it clearly. It is the transport channel, which I'll explain further, and it is surrounded by the ring. What this ring called as? This ring shows the cytoplasmic and the nucleoplasmic rings. These are the two rings which we'll see in detail. And similarly, this E and F part are showing the three-dimensional surface texture of the NPC, emphasizing their raised ring shape on the nuclear envelope surface. Sorry. Very sorry. So the NPC is often described as looking like the donut shaped from the top view and cartwheel shape from the side view, like depending on the view and image technique, that how you see it, as it has a ring with a central hole like a donut. And similarly, side view cross section through the envelope shows the spokes of the cartwheel corresponded to the radial protein subunits which connects its outer and the inner rings at the nuclear and the cytoplasmic surface. It anchored within the nuclear envelope at the site of their fusion between both the inner and outer nuclear membranes, which proves that how the NPC is a 3D structure, 3D channel that extends through both the membranes. It is not just like 2D flat ring, but a 3D extended form of complete ring structure. Hope all these basics are clear now. Perfect. Now, on the basis of studies of the electron microscope of NPC, structural models have been proposed. According to these models, the structure of NPC is supposed to be like a basket which fills the nuclear pore like a stopper. Look at it. This is your nuclear basket, and this one is the central plug, which is also called the transporter and the translocation modules. In between this central channel, together they act like a stopper that controls the passage through the pore. It consists of two rings. One is the cytoplasmic ring, which faces the cytoplasm, and it anchors the cytoplasmic filament, which is extended into the cytoplasm, that faces the cargo protein coming from the cytoplasm. These cytoplasmic filaments recognize and guide the proteins with the nuclear localization signals. And another is the nuclear ring, which is attached to the nuclear basket, and again it is extended into the nucleoplasm, which helps in the export process, in the RNA export and regulating their transport directions. Both the rings are connected with each other by the means of the spoke complexes, and together they act like the scaffold, central scaffold. And this basket-like structure is present towards the nucleoplasm and is suspended by the means of spokes. The NPC structure resembles to the basketball hoop and the net. The rings are the hoop and the basket is like the net, which is hanging inside the nucleus, and the central plug acts like the stopper that can block or release the ball. Similarly, the opening and closing of the pore controls the movement of the molecule, just like a player controls the ball through the hoops. Now imagine there is a basketball hoop with the soft nets, and there is a player's hand which acts like the stopper. This is the temporary stopper blocking the hoop. When the player moves the hand, the ball cargoes can pass through it, and when the hand covers the hoop, nothing can pass through it. Similarly, the central plug is in the middle of the NPC, blocking the passage when transport isn't occurring. And when the any molecules RNA protein has the right signal, an NPC opens up. The stopper moves and the cargo passes through the central channel. As simple as that. These nuclear pores are passive, through which the movement of RNA and protein molecules takes place in both the directions between the nucleus and the cytoplasm. There is one central channel through which the proteins and the RNA are thought to be passed, which is rich in NUPs. See, among all these nucleoporeins, those proteins which lines the central channel of the NPC possesses a large number of FG-NUP repeats. It stands for the phenylalanine and glycine repeats. It is written as FG by their single letter codes. This FG domain form a hydrophobic sieve which blocks the diffusion of larger macromolecules, which is larger than about more than 40,000 daltons, between the nucleus and the cytoplasm. Now each NPC is composed of dozens of different proteins in it. Well, it is considered to be as 30 different proteins to be precise, and they are called as the nucleoporins or NUP. These NPC is not a static structure since many of its protein components are replaced with the new copies, but overall you can say it contains only about 30 proteins in it. Now these FG repeats are clustered in a particular region of each molecule called the FG domains and makes a discordant structure that gives them a more extended and flexible organization to the complete structure. And the FG repeats containing the nucleoporins that lines the central channel of the NPC with their filamentous FG domains extending into the heart of the central channel, which is in the middle, that acts like the selective barrier which allows the small molecules for the free diffusion. Molecules below 40 kilodalton like ions, metabolites and the water molecules, they can pass by the passive diffusion, as the low molecular weight solutes are believed to pass through the slots between the spokes. And the FG domains form a hydrophobic sieve which blocks the diffusion of the larger macromolecules more than 40 kilodaltons like proteins, RNAs and ribosomal subunits. These require the energy dependent transport but not via the protein pumps. Mark here, rather not via the protein pumps, rather through the receptor mediated transports. Using what? Using the GTP hydrolysis, not the ATP hydrolysis. You will learn about these important and exportant transport receptors and their cargo type in the higher classes. For now, this is enough for you all to remember. So overall, it is highly regulated form of transport, and you can assume a 40 kilodalton as an average for the entry and exit of any kind of molecules within it. It is flexible enough to let small molecules pass through, but restricting enough also for the large ones unless they are carried by the important receptors. Look at this diagram. Can you see this flexible unsaturated region? This is the actual FG nucleoporin region. Look at it, how it links the central channel. Central channel is like a hollow cylindrical tunnel which has a plug-like structure in the middle. Look at it, the densely stained region in the middle of the central channel. This is your central plug. This is nothing but looks like the switchboard plug, a typical switchboard plug. It appears to open and close dynamically, just like the turning a switch on or off to control the flow of the current in a switchboard. Unlike these two, cytoplasmic ring and the nuclear ring, there is one more inner transmembrane ring, or you can say membranous ring, or the spoke ring in between of these two. It holds the entire NPC firmly within the nuclear envelope, so it doesn't drift or detach with each other, by providing them the mechanical support to the pore, by keeping it stable and functional. All these three rings are parallel with each other. And yeah, you can also check out these NPC electron micrograph cytoplasmic basket structure from their top view and bottom view. These are the clear image of the NPC. Both are the 3D structural models, computer generated models to help to visualize the structure of NPC more clearly. You can see the rings here in the uppermost part in the red color. These are the cytoplasmic rings, and in the blue color in the middle you can see the transmembrane rings, and in the bottom you can see the blue basket like structure. That's what? That's your nucleoplasmic ring. Perfect. And look at it, these are the green curved arms extending outwards, represents what? Answer with me. Cytoplasmic filaments, yes. And these are the yellow case like structures below. These represents the nuclear baskets that extends into the, repeat after me, nucleoplasm. Correct. So yeah, that is it for today. Thank you everyone for being till the end. Hope you all liked the lecture. Make sure you are writing all this down in your notebook. It's an important way of learning.
go through your today's notes again and revise it again and again. Highlight the important terms and it will really help you during your exam sessions. So that is it for today. Keep learning, keep smiling.